Okay, good afternoon. And today we'll start our last class, which is on continuing the role of the ventral hippocampus in behaviors that are not spatial memory. So we talked a lot about how the ventral hippocampus had a role in anxiety type behaviors. And today we'll explore that further and also look at other roles that it has in appetitive behaviors like eating food or drug consumption. Okay, so this is a paper from discussion section this week, and we'll go fairly in depth into this, both the discussion section and here, because it's one of the most modern papers on the role of the ventral hippocampus, and then you can really appreciate the complexity of using many different types of techniques to really delve into how a circuit controls a particular behavior. Okay, so the first thing the authors did was to see how the ventral hippocampus was activated during exploration of these anxiety tasks. Because previous studies had shown, is the volume too loud or is it okay? Okay. Because previous studies had shown that lesioning the ventral hippocampus caused a decrease in aversion of the open spaces, and mice don't like open spaces because they're more vulnerable to predators and such. And then the other study we talked about from when I was in graduate school showed that it looked like the ventral hippocampus was communicating with some other regions during anxiety. So all of this suggested the activity of this region would correlate with how the mouse is doing this anxiety-related task. So to look at that, they expressed the calcium indicator GCAMP in glutamatergic cells in ventral hippocampus. And then they tried to see when are these cells active. So first they started with cells in general, which are just synapsin expressing cells, just cells that have synapses, so both glutamatergic and GABAergic cells, and later they'll look at the GABAergic cells separately. Okay, so are people familiar with how this miniscope method works? This Professor Wassam covered this at all? Yes? Okay, so in case you don't remember, it's a method just to look at the activity of single cells. So you can make sort of a movie of fluorescence, and the GCAMP is becoming more or less fluorescent cells that are more or less active. And then you can plot these traces over time, like this one here. And each little peak you see is when that little cell was more electrically activated. So it has more calcium in it, and the calcium makes the GCAMP fluoresce more. So those 5 traces are traces of cells in the ventral hippocampus while the mouse was walking around in this plus maze. Okay, so, and the cell segmented field of view are the positions of the cell bodies that were extracted from this movie of fluorescence. Okay, so in panel B we have this scheme of the plus maze. So there's these closed arms where it's safer, and the open arms where it's potentially more dangerous. And then they plotted the average activity of cells as the distance from the center of the maze. See, so that red dot there is the center of the maze in between the 4 arms. And then they plotted what's the average activity for each cell when the mouse is at different distances from the center. And we see here that they plotted this in a color plot. Okay, so in the x-axis is distance from the center, and then the y-axis, each row corresponds to 1 cell. Okay, so if you look at the, say, row number 1, cell number 1, you can see that there's more yellow in the closed arm. So that cell number 1 is more active when the mouse is in the closed arm, and then it's more blue, which means it's less active when the mouse is in the open arm. However, that sounds more like an exception. If you just look at the distribution of yellow just by eyeballing it, you'll see there's kind of more yellow or more higher neuronal activity in the open arms and not in the closed arms. So although there are some cells that are more active in the closed arms, the majority of cells seem to be active more in the open arms. Okay, so any question about how they display this data here? Yeah.
So they're measuring the CA1 neurons here.
Yes.
Okay.
Yes. Yeah, all of their studies in hippocampus are ventral hippocampus area CA1. Okay, so first finding is cells appear to be more active near the open part of the environment than the closed part. And then they looked at it in more detail. So not just are the cells more active in the open arms, but how does it look like when the mouse is actually making crossings from the closed arm to the open arm? The mouse is, going from the closed space to the open space and going back and then peeking out again and then going to the open again. They're doing a lot of exploration in this environment. And here they have some example traces of what happens when the mouse makes this trajectory of going from the closed arm to the open. So he starts in the closed arm here at minus 10 seconds, and then at second 0 he enters the open arm, and then he continues into the open arm up to second plus 10. And you see that most of the cells show higher activity, which are kind of more peaks, which represent higher GCAM fluorescence, when the mouse just entered or is about to enter the open arm. So these are kind of example transitions. And they also quantified that and showed that the number of calcium transients or peaks of this GCAM fluorescence is higher in the open arm than in the closed arm. So further quantifying that initial effect. So cells seem to be more activated in the open area. And then they looked also at the other sorts of transitions, like what happens when the animal goes from like closed to open or open to close, etc. So for each little row, you see there's a number 1, 2, and then that transition is what plotted in the color plot. Okay, so the first panel there, the number 2 is in red, which is a transition from closed to open. And then you see that there's kind of more blue in the closed, and then at 0 the transition happens, there's more yellow afterwards. And they showed that this is happening when he goes from either closed arm to either open arm. So it's not just from one closed arm to the other, right? Because there's 2 closed arms and 2 open arms. So here they're plotting, okay, what do the activity looks like when it goes from closed arm 1 to open arm 1 and from closed arm 2 to open arm 2. And then in both cases, you see that there's higher activity in both open arm 1 and both open arm 2 during those transitions. And then on the second part there, they show what's happening when they go from open arms to closed arms. So in this case, they're going from the more threatening place to the safer place. And then you see that the activity goes from higher in the open arms to lower in the closed arms, which is why in the second row, the yellow is in the left. And then there are these little red moments in the second row called head dips. And has everyone heard of what these head dips are? Okay, so the head dip is sort of a exploratory movement that the mouse does to evaluate what's out there, what's the risk. So remember, there is this closed arm, which is like there are these walls enclosing the closed arm. So there's kind of a wall here. And then he goes from the open arm. And the open arm is like an open platform, kind of like just a plank extending out there. And then he goes through the open arm, and that's where it's more dangerous because things could get him and eat him from above. And then when he goes to the open arm, he goes off into the edge like this and looks to see what's there. Like, So it's like if you were at the edge of like some precipice or something, and then you just go like this to see what's. And so you're evaluating what's there, is it risky or not? And then that's a position where the mouse is under even higher risk than just being in the open arm. Because here I'm in the open arm, but I could run quickly to the closed arm. But if I'm here in the open arm and I'm doing this risk evaluation thing like this, I'm kind of hyper vulnerable because I can't even quickly run back because I'm all extended here over the edge. But they do this to kind of evaluate the risk and see is there something down there to be afraid of, etc. But in those high risk moments, you see that the activity becomes even more yellow. So it's like the activity is already higher in the open arms and is even more higher during these head dips, which are these situations of extra potential risk. So far it seems like the activity in the ventral hippocampus does seem to track well the amount of risk that the animal is exposed to. And then they show third column here, third row, what happens when they go from arms of the same type. So when he goes from one closed arm to the other closed arm, or from one open arm to the other open arm. So you see when he goes from closed to closed, the first row of the third panel here, it's all blue because the activity is suppressed in the closed arm and is also suppressed in the other closed arm. And when it goes from open arm to open arm, it's high activity all the time because they're both open arms. Okay, so the reason for looking at all these transitions from one direction to the other, to the other, to the one, is to really be sure that the cells are indeed more active in the open arms. Because like this result here, if you just looked at this, okay, it looks like there's higher activity when he goes into the open arm, but perhaps there's just higher activity when he goes in, enters any arm. Like maybe if he enters any arm, there's high activity, and then the activity decreases, then he enters any other arm, there's high activity again. So this could be explained by that. But if you look at all these transition possibilities each at a time, you can see that, oh, it's not that there's just high activity when he's moving or high activity when he enters any arm. There's high activity specifically when he enters the open arm. Okay, so then they try to make some controls for it because the plus maze is sort of a novel environment, right? So he has his home cage, which he knows and is very happy there. And the plus maze is other places he's never been to, so it's very novel. And in mice, kind of novelty and anxiety-provoking stimuli are always intertwined because the mouse is typically not scared of very familiar things that he knows very well and nothing bad ever happened there. So that makes sense. So, but one confound of the plus maze is that it is novel. So perhaps this activation they're seeing is because maybe it's a novel environment. That's what's going on. Okay, so they expose mice to this novel object task, which is basically you have a box, and inside the box there is a place that's empty and this other place that has a novel object. So the mouse is very curious with the novel object. It's like, oh my God, it's a novel plastic object. Let me look at it, let me rear against it, let me smell here, let me go for the other side to see what's happening. So they're very curious with this novel object. But the novel object is not scary. And then you can see, he explores more the novel object than the neutral empty place. But importantly, the ventral hippocampal activity is not higher in the novel object. So this shows the results in the plus maze are not explained by simple novelty. Okay. And then they do this very cool thing here, which is trying to see if the neural activity patterns they saw across mice correlates with each mouse's individual tendency to be more or less anxious in this maze. Okay, so what they plot in the x-axis is easy to understand, which is how much each mouse explored the open arms. So mice that are more in the left were more scared of the open arm. Mice that are more on the right explored the open arms more, so they're presumably less scared of the open arms. So that's the x-axis. The y-axis is the average difference of ventral hippocampal cells for a particular mouse of the activity rate in open arms minus activity rate in closed arms. Okay, so what that means is that, let's erase. Okay. So let's say this is the open arm and this is the closed arm, and one particular mouse perhaps had like a lot of activity in open arm like this and like no activity in the closed arm. So in this mouse, the difference in activity between open arm and closed arm is very large. So this mouse would have a high y-axis point there. So basically what they're showing that mice like that had a very high difference in ventral hippocampal activity, those mice also explored the open arms less. And then you had some other mice where maybe the open arm had like that much activity, and then the closed arm had like a little activity. So for this yellow mouse, there's less difference between the activity in the open arms and closed arms, and those yellow mice also were less scared of the open arm. They spend more time in the open arm. So here they're doing this cool thing, which is correlating across individual mice their neural activity results. Because the neural activity is related to the behavior. Not only would you see that in the open spaces and closed spaces there should be some difference, but also mice that individually behaved more or less anxiously should have some difference in that metric as well. So they also find that. Okay, so any questions about how this graph worked? Yeah. Open arms. Yeah, because the closed arms are closed, like there's a wall here, so I cannot, I'll hit here that wall, so I can't head dip. So the head dip is kind of here, like in this open ledge, which is open, and then I stretch my body like this and look and see, and so I'm dipping my head beyond the edge of the open arm. So it sort of can't happen in the closed arm. So the closed arm is here, so he would hit the head, and I couldn't head dip. So they do that to see what's happening. Is it very scary or not? What do they do? And you're like, why do they do this? It's kind of more risky. But you also do that in some risky situation. You know, if you were told like, oh, there's some assailant that's coming here, like, then you wouldn't spend all your time hiding. You would also like, is there? Is there not? Let's see. You do that kind of risk evaluation as well. Okay, so, okay, yep.
So if I'm understanding, the more activity we see in the open arm, the more time we're going to spend in the open arm? For graph G?
No, the opposite.
Okay.
So, more higher activity in the open arm would be bigger y-axis position. So those have lower exploration of the open arm. So mice that in which the neural activity was very different, like very high in open arms and very little in closed arms, those mice also avoided the open arms more.
And for graph D, are they recording from one neuron for all three parts?
No, these are averages of many neurons. Or no, sorry. These are they're example traces from representative neurons. These are not averages. Yeah. Yeah.
So for 2 slides ago, when you showed with when you showed the fluorescence of the ventral CA1, and also there are some like correlations within the CA3 to for the phosphorylated GF3 there. Is it also correspond to the ventral hippocampus that also active during the open months? Or is it just the CA1 that you were
This is only CA1.
Well, why is that like a fluorescent?
Oh, that's because even if you inject the virus in CA1, then the cell bodies at CA1 take up the virus. The cells have all these axonal projections that go to various places, and those would become green also. But the imaging was done where the cell bodies are, which was the CA1.
But like for the, it's encompassing all the CA1 regions, not just only the ventral CA1.
Oh no, this is only ventral CA1.
Okay.
Yeah, this is only ventral CA1. Yeah, the dorsal CA1 is not even in this section. It's kind of more anterior and higher up, like more dorsal.
So whenever you say about ventral hippocampus, it's directly recording in the ventral CA1.
Yes. Yeah, so far everything is ventral CA1. Yeah.
Did you mention, like 2 slides ago, that it's single neuron recordings?
Yes. Yeah, so each trace here is the kind of fluorescent trace of 5 individual cells. And then they record that many cells as shown in the field of view. So each little circle there is 1 cell. So it looks like something like 100 cells or so were recorded simultaneously. Yep.
Professor, may you please restate the significance of the novelty item?
Mm-hmm. Oh yeah. So One confound in this plus maze experiment is that the plus maze is by necessity novel, because if it's a very well-known thing and they live there and they've been there for hundreds of hours, then it wouldn't be necessarily scary. So it has to be novel. But then it has now two features. It kind of has these open spaces and it's novel. So they want to show their effects are not related to the novel part. So to look at that, they did this experiment in E, which is they exposed the mouse to a novel object, like this bottle of water. And it's an object they've never seen, so they're like, wow, it's a bottle of water, look at this, it's the best thing ever. And then, but they're not scared of the bottle of water, but it's a novel thing. And then they show here that the cell is not more active near the novel object compared to empty area. So it shows that the effects in the plus maze are not explained by novelty, they're better explained by openness, open space. Yeah.
For like the G1, what that means with the graph is that the more exploration time you have in the open arms, the less activation on the CA1 activated the ventral hippocampus.
Sorry, say again?
Like the more exploration time that you have, the less activation of the CA1 in the open arms.
Yes.
And then does that mean like the mice is not actually scared when it goes to the open arms, or like?
It suggests they're less scared because they're going there more.
Oh.
Yeah, but they're all reasonably scared, because even this mouse that explored the open arm more is still spending 30% time in the open arm, not 80% time. So even the brave mouse is still more avoiding the open arms than not. So they all avoid it. Yeah.
So with the head dips, since it's firing more after the head dips, does it mean that the firing would kind of signal danger?
It-
Not the opposite?
It would suggest that more, so far, that more dangerous situations create more activity, yes.
Can we discuss like the context? Last time we talked about it could be that they're more brave, they could be like less scared.
Oh yeah, so regardless of whether the mouse is more brave or less scared at that in the open arms or closed arms, he's definitely more exposed in the open arms than closed arms, regardless of his internal psychology. And then when he is in the open arms, he's maximally unsafe when he's even more exposed doing this head dip. So, and that we can be pretty sure about, like that he's more protected in the closed arm. But we can't be super sure about, is he more scared when he's in the closed arm. So these results, you could say, show a good correlation between the ventral hippocampal activity and exposure to risk or potential threat. Yeah.
Why is there a speck of yellow in the first one, like open to closed, like in the closed section? Like it seems like fires,
Oh, that's because they were honest and put a representative trace. So it's like, it's not that there's zero activity in the closed arms, there's just less in average. But they could have selected like example tracers, there's like no activity whatsoever in the closed arm, like enormous activity in the open arm, but they chose one that kind of, there's less in the closed arm, but not zero. Yeah, okay, any other request? Okay, so they did these things, then they wanted to see, okay, so these are all cool correlational data, so now let's see what happens if we inhibit this region. So they inhibited this region using the inhibitory opsin arch, and now they are inhibiting the glutamatergic cells specifically because use this virus that infects glutamatergic cells. Okay. And they didn't just inhibit it in the plus maze in general. They were super elegant and inhibited it in a way that was informed by the neural activity recordings. Okay, so what did neural activity recordings show? The neural activity recordings showed that these cells are more active in the open arms. So if they have more activity in the open arms, that's the moment that it makes sense to inhibit them. Because if you inhibit them in the closed arms, they're already inhibited anyway, so what are you inhibiting? So they didn't inhibit it just when the mouse is in the plus maze, they separately inhibited only when the mouse was in the open arm or when the mouse was in the closed arm. So the mouse is doing its own thing. I'm going to the open arm, I'll go back to the closed arm, I'll stay here, now I'll go back here, then I'll go to the open arm again. And so they did 2 different experiments. In one experiment, they inhibited the region whenever the mouse went to the open arm. And in this other experiment, they inhibited whenever the mouse went into the closed arm. Okay, so when they inhibited when the mouse was in the closed arm, which is the second column, nothing happened. And that's not super surprising because if you look at this thing, the cells are essentially not very active in the closed arms. So if you inhibit this moment that they're already not active, there's not much to inhibit, so not much happens. But if you inhibit in the open arm, which is when there is more activity to be inhibited, then that increases the amount of time they spend in the open arms. And this kind of replicates their correlative data, right? Because here what they're doing is, if you inhibit activity in the open arm, so there's less ventral hippocampal activation in the open arm, that makes the mouse stay longer in the open arm. And this is a causal manipulation experiment, right? And this recapitulates their correlative observation, which is that if we just look at the neural activity and don't manipulate it, its natural variability is such that mice that spend more time in the open arms have lower activity in the ventral hippocampus. And then they show the exact same thing causally, which is that if we now force the activity to decrease, mice spend more time in the open arms. So it's a very cool convergence of the recording and manipulation data. Okay, so any question about this part here?
Can we say that the ventral hippocampus is some sort of like warning mechanism of like, hey, you shouldn't be here right now, and so it goes to the closed arm?
Yes, that's a possible interpretation of what's happening.
I see. Thank you.
And this also is an experiment that helps the interpretation. So here they did this experiment called real-time place preference. So it's basically an environment divided into 2 halves, like let's say here and this area. So whenever the mouse goes into this area, there is optogenetic inhibition. And whenever the mouse goes into this other area, there is not. Okay. So the reason they do this is to figure out if any effect is being caused psychologically by this optogenetic manipulation in this neutral environment. This is not an anxiety-provoking environment, it's just a random box with 2 halves. Okay, so it's not about this experiment, but imagine that in this real-time place preference, whenever I go here, they stimulate some panic-inducing nucleus in my brainstem. So then what would happen is I'm like, I'm here, it's very good, and then I go like, oh my God, it's the worst again. So I'll go back here. Okay, so if that were to optogenetically happen in me, that some terrible experience happens because I panic when I go here, then I would avoid going there. Or conversely, if something nice happened, like they stimulate some pleasure nucleus in the brain, or they infuse cocaine in me or something like this, whenever I go here, then I'll be like, oh, cocaine is so good, I'll stay more time here. So it's basically a way to see if the manipulation is liked or disliked by the mouse. Now, this does have some caveats, because it could be the mouse stays here longer, but it's not because it's more pleasurable. Perhaps it's just that stimulation reinforces the current action being made, and then I'll just make more of this action, which is staying here, even though it's not pleasurable. So there's some caveats in the interpretation, but it is an easy way to see if the manipulation is causing aversion or some kind of approach positive motivation. And here they see it doesn't cause that. So this manipulation on its own doesn't seem to, for example, decrease the baseline pre-existing levels of anxiety in a neutral environment. So that's what they show here. Okay, and then they went on to see what is the amount of cells that are more active in the open and closed arms. So now they recorded also from the dorsal hippocampus, which is our friend from the first week that we haven't talked about for a while. So the dorsal hippocampus, if you remember, is important for spatial memory and navigation and episodic memories and things like that, and not important for anxiety. So if you lesion the dorsal hippocampus, the mouse does not change its anxiety behaviors. And then they record with the miniscope in the dorsal hippocampus in the plus maze, and then they see in average there's not more activity in closed versus open arms in the dorsal hippocampus. They don't particularly seem to have a preference of arm type in average. And in ventral CA1, as we said before, there's more activity in the open arms in average. So there are like 50% of the cells are more active in the open arms, and only 19% are more active in the closed arms. So way more cells are active in the open arms, preferentially active in the open arms. And the dorsal hippocampus kind of split half and half. Like there's not some strong preference of cells firing more in the open or closed arms. And that makes sense because the dorsal hippocampus is not really concerned with anxiety. It's concerned with representation of space and timing and experiences and things of that type. Okay, so any questions about this part here? Then they do this cool control to show if those cells that were more active in the open arms of the plus maze, were they also active in some other open space. And this is very cool and important because ideally you'd want to see in any of these papers that claim this neural activity encodes a certain kind of state that it's doing that at least in two different occasions, right? So to do that, they run the mice in the elevated plus maze and record from ventral CA1, which is the first thing here. Then they select the cells that fire more in the open arms of the plus maze. And as we saw, 50% of the cells fire more in the open arms. So now they look at that subset of cells that fire more in the open arms of the plus maze. And because they're selected as being cells that fire more in the open arms of the plus maze, they fire more in the open arms of the plus maze, like as a circular consequence. So not super exciting. But then something interesting happened, which is they look at those same cells that fire in the open arms of the plus maze and put them in the open field, which is the different paradigm which has open spaces and it's a different box in different place, etc. And they see that cells that are more active in the open arms of the plus maze are also more active in the open center of the open field, which is a completely different box. So what they have in common with the plus maze is the openness. Because it could be that the cells that fire more in the open arms of the plus maze, maybe they're not firing due to openness. Maybe they're firing because there's a weird smell in the open arms. Or maybe they are firing because in the open arms there's like a wind draft that they can feel, and that's very nice. Or maybe it's like more brightly lit. Like it could be also, maybe I can see more interesting things. Like there's all sorts of things that are different between the open arms and closed arms, which are not the openness exactly. And you can't control for all those things. So what you can do is to see if the cells also respond to this other open space in this other location, right? So, and that's a cool control. So it's something, let's say if you were studying a person and you see like, oh, this person likes to hear this song by the Rolling Stones. And from that you should not conclude this person likes rock. Like you should, because maybe they just like any noise. So you expose them to some like other source of noise, like no, I don't like this one. And then you expose them to another similar thing, like a Led Zeppelin song. Then like, oh, I like this one too. And then with like two observations, you are kind of more certain that they were liking the rock part of the song and not it's a noise of some sort. So here too, they do that, and the commonality between the open arms and the open center of the open field is the openness. So with this, you can very confidently sort of say these cells are responding to the openness. And then they also show that the open arm preferring cells are not more activated by the novel object. So the same control you saw before. And then they do the same thing for the dorsal hippocampus. So the dorsal hippocampus cells, which is here, they have some cells that are more active in the open arms. But that's not unexpected, because if you remember, the dorsal hippocampus has those place cells that are, and different place cells are active in different places. And some of them will have place fields in the open arms. So some cells should be more active in the open arms. Okay, so they find some dorsal hippocampal cells that are active in the open arms. And then those cells fire more in the open arms because they're selected as being cells that are more active in the open arms. And then those cells are not more active in the open center of the open field. And they're also not responding to novelty. So here the cool thing is they're showing that the dorsal hippocampal cells that are firing to the open spaces, they're not really firing to openness. They're just firing in that like GPS location kind of. And in this other environment, they don't care about the openness. So here it would be an example of these open arm preferring cells of the dorsal hippocampus are not really representing openness. They were just representing that random patch of space which happened to be in the open arm. Okay, so any questions about this part here? Okay. Yeah.
The elevated plus, that's just basically another word for the maze.
Mm-hmm.
Yeah? Thank you.
Yeah, okay. So these were recorded in glutamatergic cells. So then they see, okay, what about the GABAergic cells? There are also GABAergic cells in the ventral hippocampus, so what are they doing? Then they express the GCAMP virus in the GABAergic cells and record their neural activity, and they can get some traces with some nice fluorescence transients. And they see that the GABAergic cells are, on average, not more active in any particular arm type. So the average rate of transients is not higher in one type versus the other. And it looks like, if anything, there are more cells that are more activated in the closed arms rather than the open. But they're more kind of uniformly firing or have a weak preference for the closed arms. Okay, so the GABAergic cells in the ventral hippocampus don't seem to have this anxiety location encoding type of thing going on. Okay, so you see like a really narrowing down compared to what we knew before, because initially we just knew ventral hippocampus was important for this open arm exploration behavior, because if you lesion the whole region, then they lose their aversion to the open arm somewhat. Then they show, oh, cells of the hippocampus, ventral hippocampus seem to be active in openness. Then they go, but which cell? Oh, the glutamatergic cells do seem to care about this openness, but the GABAergic cells in that region don't care about this openness. See, so now we have that initial observation of the ventral hippocampus was important for anxiety type behaviors, and now we can make a much more refined statement that glutamatergic cells in the ventral hippocampus seem to care about anxiety. Okay, and then they try to see which long-range axon terminals of the ventral hippocampus to different regions might be creating these effects. So they inject a virus in the ventral hippocampus and they see, where do these cells project? Where do these axons go? Where do they end? And then they see that there are axon terminals visible in the basal amygdala or basal medial amygdala, BMA, and also in the hypothalamus, the lateral hypothalamic area, LHA. And then they kind of quantify the amount of green fluorescence in those places, and both of them have a lot of green fluorescence. So those are places that these ventral hippocampal cells project to. Okay. Then they try to see if the cells are releasing glutamate or not, and they should be releasing glutamate because they injected this virus that infects the glutamatergic cells. So it's expected that these cells are glutamatergic, but they kind of verified that by doing a slice patch clamping experiment. So here they injected first channel rhodopsin in glutamatergic ventral hippocampal cells, And then those cells produce channelrhodopsin. Then their axon terminals also produce channelrhodopsin. Then their axon terminals go to faraway places like the hypothalamus and amygdala. And there are little axon terminals from the hippocampus that go to these regions that have now channelrhodopsin in them. Okay, so this is something that's technically very surprising to think of it, because they make a brain slice that contains this basal amygdala, which is this region here, basal amygdala, so it has some axon terminals. But there's no ventral hippocampus in this slice. There's just the end of the axon terminal, right? Because the ventral hippocampus is like here, the cell body of the ventral hippocampus. Then it sends an axon terminal that goes some far away place, like here in the hypothalamus or amygdala. And if you make a slice just of this place, it just contains the axon terminal. It doesn't contain the cell body, which is all the way here in the hippocampus. And that slice of the brain containing the amygdala and the channelrhodopsin expressing axon terminal from the hippocampus is still sort of functional. Like that little dismembered axon terminal with no cell body attached to it, it still kind of works, which is somewhat surprising and cool. So if they patch clamp a cell in the amygdala and stimulate those dismembered axon terminals, they still do their thing, release glutamate. And you see a excitatory response there, and the response is blocked by glutamatergic blockers, which are AP5 to block NMDA receptors and NBQX to block the AMPA receptor. So you see the response goes to essentially 0 if you block glutamatergic transmission. And the same thing was observed in the lateral hypothalamus. So these ventral hippocampal cells send glutamatergic projections that release glutamate onto the hypothalamus and amygdala. So that's what's shown here. Okay, so any question about this experiment here? Okay, so now they want to see, do the same or different cells project to amygdala and hypothalamus? And the way they do that is by injecting a retrograde tracer of different colors in the hypothalamus and amygdala. And the tracer they use here is CTB, which is short for cholera toxin subunit beta. And cholera is like a very terrible disease that has killed like millions of people, and the kind of microorganism that causes cholera has this toxin called cholera toxin, and then this cholera toxin is a protein that naturally exists that is able to travel retrogradely, meaning if you inject that protein here, then it's taken up by axon terminals that are nearby here and then travels along the axon and infects the cell body of cells that project to that place where you made the cholera toxin injection. So there's a lot of kind of microorganisms that kind of infect people that have this ability of traveling retrogradely across axons, and that's because typically the infection doesn't happen in the brain. The infection usually happens in some other place, like the periphery of the body, and then to gain access to the central nervous system, the microorganisms have to figure out how to travel retrogradely. So say if you get rabies because you're bit by a bat or a rabid dog or something, the dog is usually not biting you in the brain. The dog generally bites in the leg or something, and then in your leg you have like axon terminals that come from the spinal cord. So the rabies virus infects those axon terminals, then travels all the way to the axon and goes to the spinal cord. Then from there it infects the axon terminals that innervate the spinal cord, and then it reaches the brain. And then you become rabid and start biting other people. So that's why there's lots of things that microorganisms do that allow them to travel retrogradely in the axon terminal. So they use this CTB technology, and they inject CTB from the cholera toxin that is covalently bound to either a green or red fluorescent dye, and they inject those things in basal amygdala and hypothalamus, and those are taken up by axon terminals of places that project there. And then if you look in the ventral hippocampus, you see some cells that took up these retrograde tracers, because they project and send axon terminals to those places. And they now take up this green and or red color that you injected in the postsynaptic regions. Okay, so one cool thing that you see is that the green and red cells appear not to overlap very much. And they also appear to be sort of in different subsections of the cell layer. So this suggests that there are different cells in the ventral hippocampus that project to amygdala and hypothalamus. Because if the same cells projected to both regions, then the same cell would become red and green, which would show up as yellow in this colorization. And there are very few yellow cells. There's a bunch of red and a bunch of green cells, suggesting that different cells project to these different places. Okay, so any questions about this one? Yeah.
So they use two different retrograde tracers?
Mm-hmm.
Okay.
Yes, of different colors injected in the same mouse. Right, so now that we know there are different cells, they wanted to see what does a projection to amygdala do. So they injected a virus in the ventral hippocampus, then there's axon terminals carry the CHR2 all the way to the synaptic context in basal amygdala, and then they do contextual fear conditioning, which is a behavior that is known to require both the ventral hippocampus and the amygdala. So what they do here is they do the fear acquisition first, which is the first panel, while having optogenetic stimulation. And then they test it the next day by putting them back in that same box and without any optogenetic stimulation. And they see that the mice that had received optogenetic stimulation on the training day now freeze less. So activity in this projection seems to be important in the acquisition of the fear conditioning training. Then they do also train normally, so in the shocking day, no optogenetic manipulation. And then the testing day, if you optogenetically excite those terminals, you see less freezing, showing this projection is also important for both acquisition and retrieval of the contextual fear memory. Okay, but here though, there's something very strange happening if you think about other things we learned in class, because here we are exciting the projection, so more neural activity, and you get sort of less freezing, right? But if you remember the other stuff we talked about earlier, we said that lesions of either of these regions cause less freezing in contextual fear conditioning. And lesioning is kind of less neural activity, right? Because the cells are not there because you killed them. So this result seems to be at odds with those other results, because here we have activation causing less freezing, and those other results showed that inhibition also causes less freezing, because lesioning is kind of a type of inhibition, right? So this was very perplexing. And then the explanation they gave is that the optogenetic excitation they're doing perhaps was just adding noise to some very careful time sequence of neural activity action potentials that were happening, right? So what's likely happening is that to learn contextual fear conditioning is not just a matter of you need activation in this circuit, but you need some precise activation that carries some important information. And then if you just optogenetically blast it artificially in some strange way, that kind of disrupts that internal information, so you end up getting loss of function. Okay, so one way to think about it is, let's say you have like some antenna and you're receiving some information from a radio station, and now you can play this song here. So there's some information coming through these radio waves, and the radio can get that and play this song. Okay, so if you do a lesioning experiment, which is you say destroy the radio station, then the song will disappear, because there's no information being sent to the radio. So the lesioning will produce worse music, which is less music. But if you just added a bunch of radio waves, which is kind of more radio wave, that would also produce less music, because the music is not just, there are some radio waves, but it's kind of a very precisely structured radio wave that carries that information of how to play the music. So adding random noise to the system, which is kind of what they're doing here with the optogenetic activation, would also potentially just decrease the information transfer. So that's why they said it decreases the freezing in this case. Yeah.
So when it comes to this activation, do you know, like temporally how it works? Was it activated only during the presentation of cues, or was it like a blanket during the session activation?
It seems like it's activation during the session from the scheme, because the blue light is not when the thunder occurs. It just seems like it's active during the presence of being in the box. Yeah.
So if I'm understanding, shining the light during training and testing is decreasing the amount of freezing we see?
Mm-hmm.
Okay.
Yes. So you had a question?
Yeah, I was gonna ask, does this mean that maybe it's like overactivation? Like this is not
Yeah, it's likely a incorrect type of overactivation, right? Because this suggests that there is some, it's not just a matter of more action potentials in this pathway, but there's some precise sequencing of timing of when the action potentials have to happen from different cells. And optogenetics, you're just making all the cells synchronously fire at like 10 hertz, which is a very strange state of affairs. And that's why you don't get the behavior. But the converse of this is that, we have seen a lot of papers where the optogenetic activation does cause more of the behavior. And it suggests that in those many experiments, the precise timing of when the action potentials happen is not super important to cause the behavior. Right? Because if you have this nucleus that causes, say, like more eating, which is something that Professor Wassam showed, then if you inhibit causes less eating, and inhibiting is easy to interpret because that activity is not there. The excitation is you're adding some artificial activity, and then that causes more eating, right? So that suggests that in those cases, exactly when and what the cell was doing to be active wasn't super important to cause that more eating. Yeah?
Easier explanation just be that maybe these neurons eventually synapse on the inhibitory neurons, and therefore overactivation would essentially be a decrease in activity because you're overstimulating.
Yes, could be. But that too is a way of it falls in this broad category of you're activating in some incorrect way that's not doing the fear conditioning. So there are infinite ways that you could be stimulating incorrectly. Yeah, because there's only one way of stimulating correctly, which is what the cells are naturally doing if it requires some very precise organization of the timing. And everything else would be some not physiological activation. Yeah?
Could this sort of relate to theta waves in like the hippocampus? Because we know already that the dorsal hippocampus exhibits like theta waves
And the ventral too.
and the ventral.
Mm.
So could like the stimulation here be disrupting that?
Yeah, it's a very good point. It's totally possible that happens because we know from lots of other studies that the hippocampal's output is not just more active. It's kind of activated in this rhythmic fashion that when the cells are naturally synchronized. And here they're definitely not doing that. So that could be totally a very good observation that that's a way that we know is how the cell is naturally being active, and this is totally not doing that. So it could be that that's a reason why it's causing noise in the system. Okay. Any other questions about this? Yes?
Do you know if there's any inhibition studies on this specific pathway? And if so, would we expect it to have the opposite effect?
No, you expect to have the same effect.
Right. So I was going to say, because if it has the same effect, then it would be the noise. But if
Yes.
it's the opposite effect, then it could be.
It's the opposite effect, then the noise explanation is wrong. Yes, that's true. But you expect the same result if the noise idea is true. But nevertheless, it does somehow implicate this pathway in fear conditioning being noise or not. And then they showed that activating. Sorry, yeah.
On the previous slides, when you show about the training with the light on, what does it conclude on that? Like,
Oh, it concludes that the association between this box and this box has shock requires some kind of activity of this circuit. So yeah, the first experiment is testing the role of the circuit in the learning the association that this box causes shock. And then the second experiment is retrieving the association I learned yesterday, so I'll freeze now.
so that's like the first experiment is to show that this pathway is corresponding to the contextual fear conditioning, and then the second one is when you wanted to actually experiment using the retrieval to
Yes,
see if there's
yes, the second is the retrieval, yes. Okay, so then they also see what happens in the anxiety paradigm. So if you activate those cells, it doesn't seem to change the exploration of this open space in the open field. And it doesn't seem to produce preference or aversion in the real-time place preference test. So this projection to the amygdala seems to be somewhat specifically influencing learned fear acquisition and retrieval, and not influencing innate anxiety of open spaces. Okay, so then they do the same study for the other pathway, which is the pathway from ventral hippocampus to lateral hypothalamus. And then they see in the lateral hypothalamus, if you activate that pathway during either training or testing of contextual fear conditioning, nothing happens to the freezing. So this pathway to lateral hypothalamus seems to not be important for learned contextual freezing. But if you activate this pathway in the open field, you see that it produces a decrease in the exploration of the center of the open field. So activating this pathway produces more aversion to the open space, suggesting it increases anxiety. And then it also seems to cause real-time place aversion. So here what's happening is, when the mouse is in this place, nothing is happening. And whenever the mouse enters this other place, there's optogenetic activation of the ventral hippocampus pathway to hypothalamus. And then the mouse avoids this place. So here, and then the mouse spends more time in the place where you don't have stimulation. So whenever he goes here, there's stimulation of the hippocampus pathway to hypothalamus. So then the mouse somehow doesn't like that, so avoids that place. And then this suggests that perhaps activation of this projection just causes more higher defensive state or more anxiety, etc. So whenever the mouse is here, it's like, oh no, something will happen, someone will eat me, it's not good, I'll go back here. And we don't know that's what's going on in the mouse's head, but we do know something negative, aversive, bad, etc. is happening psychologically to the mouse to not spend time in the stimulated part of the environment. Okay, so any questions here? Then they do this very cool thing, which is, okay, we already studied the activity of ventral hippocampal cells and glutamatergic and GABAergic cells separately. So now let's look at cells of ventral hippocampus that project either to amygdala or lateral hypothalamus. So the way they do that is they inject in the amygdala this new virus that I think we haven't talked about so far, which is called canine adenovirus. And this canine adenovirus is carrying the DNA to make this protein called Cre recombinase. So you inject that in the basal amygdala, and this is a virus that also travels retrogradely, just like the cholera toxin B. And then it infects the cell bodies that project to that place. such as ventral hippocampal cell bodies that project to amygdala. So now, ventral hippocampal cell bodies that project to amygdala have Cre recombinase being produced in them. And then you inject a second virus, which is this AAV1 GCAMP, but the little flex before the GCAMP indicates that the GCAMP will only be produced in the presence of Cre recombinase. So you're injecting this virus in the hippocampus that will only express the calcium indicator GCAMP in the presence of Cre recombinase. And who has Cre recombinase? The cells that project to amygdala. So then what you end up with through these super cool tools of viral molecular technology is you're able to express this fluorescent calcium indicator GCAMP only in cells in the ventral hippocampus that project to amygdala. Okay, so here's a photo of the cells that project to amygdala, and then if you look in the amygdala, you see some terminals in the amygdala because those cells project to the amygdala, so there's some GCAMP fluorescence in the amygdala, in the axon terminals, but you don't see fluorescence in the hypothalamus, so that means those same cells that project to amygdala do not project to hypothalamus because you don't see terminals from the amygdala projecting cells in the hypothalamus. And they do the same thing to label cells that project to lateral hypothalamus, which are these cells here. And those cells don't project to amygdala. You don't see any axon terminals in the amygdala, but they do project to hypothalamus. So this is also a way that it confirms their previous result, which is this result here. This result showed that there are very few cells that project both to hypothalamus and amygdala. They seem to different populations. And then this is another different way that confirms that. Because the same cells projected to both places, then labeling one of the populations would result in axon terminals in both of the places. Okay, so they use this to express GCAMP selectively in cells that project either to one place or the other. And then they record from them in the elevated plasmase. So they get some calcium transients. And here's what they find that's cool. They find that the cells that project to lateral hypothalamus have a bigger difference in activity between the open and closed arms than cells that project to amygdala. And that's cool because, again, the optogenetics results showed that the projection to hypothalamus was important for anxiety and the projection to amygdala was not. And here we see that cells that project to hypothalamus represent the open arms more strongly than cells that project to amygdala. So nice convergence between the optogenetic results and the neural activity. And here they show that a staggering 80% of cells that project to hypothalamus are more active in the open arms. And a big chunk, which is 47% of the cells that project to amygdala are more active in the open arms, but it's like way more in the cells that project to hypothalamus. So here we're seeing something interesting, right? Because the first figure they showed that generally cells in the ventral hippocampus are more active in the open arms. But then if you subdivide them into cells that project to amygdala or hypothalamus, first you see they have different roles in behaviors. And also that the cells that project to those two different places have different amounts of preference of firing more or less in the open arms. Sorry, any question here? Okay, so here we're seeing a kind of very fine gradation of how to look at this circuit. Because it started with ventral hippocampus is important for anxiety, but now it's glutamatergic cells of the ventral hippocampus that project to lateral hypothalamus. Those cells are the ones that are particularly more important for anxiety, right? So this is something like going from saying New York is important in the financial system to saying Wall Street within Manhattan is the particular sub thing in New York that's more important for the financial system. So it's kind of increasing the resolution of the claims we can make. Right. So they also show the kind of loss of function effect, which is that if you inhibit the projection from ventral hippocampus to hypothalamus, you get more exploration of the open arm. And they also showed the gain before, right? If you activate them, you cause less exploration of the open space. So if you inhibit it, causes more exploration of the open space. So it's always good to see that the correlational measures and the gain and loss of function measures all converge in the same direction, providing like a very strong argument for the overall conclusion. Okay. So that was this paper. So any questions about that paper?
So for this ventral CA1 neurons, could you say that they're active towards like innately anxiety-inducing environments? So like they're more active during the open arm, or is there some kind of like learned fear component?
Yeah, they were more active in the opener. Yeah, well, they didn't record in the learned fear, so we don't know what was happening there, but they're definitely more active in the opener. Yeah, and the cells that projected to amygdala were also more active in opener, but less more active than the ones that project to hypothalamus. Yeah, and here we can also kind of tie in with something that Professor Watson talked to you guys about, because she also showed these studies showing that the lateral hypothalamus has cells that are important for causing voracious eating, like the GABAergic cells there, if you activate, cause a lot of eating. Now we're seeing a different thing, which is there's this projection here that causes more anxiety if you activate them. So that region is important both for feeding and anxiety. So it's like you can really parse out lots of cool functions of the same region using these different methods. Yeah.
How many like months or years of work does it take to like publish a paper that has that many experience?
Like 4.
Years?
Yeah, and it also depends on how many resources you have, how lucky you are, because it could be the first year studying some other thing and it didn't work, and then you got nothing out of that. Yeah, but these studies are like, as you notice, very technically challenging, and they're like way more challenging than those early studies we covered historically, because the first papers were kind of, if you lesion this region, the mouse does less of something, and that's the whole paper. And now it's like, if you activate this projection of this glutamatergic cell type to this other sub-region of the hypothalamus, then this one behavior happens, but the other behavior doesn't happen. So it's very tightly controlled. But then we get to learn a lot more, and then the graduate students become much more stressed and sad. Okay, so the fact that this projection to the projection lateral hypothalamus area, in the lateral hypothalamus was important for feeding, suggests that perhaps this region too could be important for feeding, because it's connected to one feeding region. And then to look at this, they tried to see if the injection of ghrelin in the ventral hippocampus causes any effect in feeding. So ghrelin is a hormone released by the gut when the gut is empty. So when you're hungry, the gut is empty, and the gut's like, I don't like being empty, I like being full of ice cream and fat. So then it releases a bunch of ghrelin into the bloodstream, which makes you hungry. And then there are lots of ghrelin receptors in the brain, in different brain regions, that are able to bind to that ghrelin that came from the blood, and then they initiate behaviors related to eating more. And the hippocampus wasn't really known to be involved in this, but it does have ghrelin receptors, so these people thought maybe it does sense feeding somehow. So they injected ghrelin inside the ventral hippocampus and saw that that increased the amount of food the mouse eats and also the frequency of meals that the mouse is taking. So this was a cool result because it shows that now the ventral hippocampus is involved in kind of learning contextual and tone fear conditioning, innate anxiety of open spaces, and also feeding when you're hungry. So many different potential roles that we don't know, of course, if these cells that produce the hunger are the same cells that produce the anxiety or not. Yeah.
Do we know what effect ghrelin has in the ventral hippocampus, whether it's excitatory or inhibitory?
Yeah, it's a good question. They didn't do that in this paper, but that would be good to figure out. Okay. Then they also tried to see if this ghrelin injection not only caused the mouse to eat more, but also does it cause some more sophisticated type of food seeking? So rather than the food is just there and he eats, would the mouse also do a more learned instrumental action like lever pressing to get food if the ghrelin is injected in the ventral hippocampus? And then they find that yes, if you inject ghrelin in the hippocampus, the mouse does more lever pressing to get food, and it presses the correct lever that gives food and doesn't press more of the inactive lever that doesn't give food, which is a good control because it shows that the ghrelin injection is not just causing psychopathic pressing of levers, it is causing specifically pressing of levers that give food. Okay, so and in the same paper they also showed that if you have kind of a not instrumental but kind of Pavlovian cued induced eating, which means there's like a auditory tone, and then when the tone stops there's availability of food here, so the mouse learns to do this. And then if you do this in the presence of ghrelin injecting the ventral hippocampus, the mouse, after hearing the tone, eats more food. So through many different measures of just free food eating, instrumental actions to get food, or kind of cued availability of food, we can see that injection of ghrelin in the ventral hippocampus causes more eating. And this goes, we don't know the answer, but it kind of is related to this question of is the ghrelin excitator or inhibitor in the hippocampus? Because it was also shown by this different group that if you just broadly activate glutamatergic cells in the ventral hippocampus, it decreases food intake. Right? And this is just cells in general, so they just decrease food intake. So it suggests that the ghrelin is not broadly exciting all the cells in the ventral hippocampus. But it could be that there's some subset that if you excite just that small subset, it causes eating or something like that. But it does, these two results taken together indicated the ghrelin is not just exciting most cells strongly in the ventral hippocampus, because this manipulation is exciting most cells strongly chemogenetically, and that causes less feeding. So you see that there's this kind of interesting dynamic where different manipulations of the same brain region can cause very different behaviors. Okay, so any question about the feeding experiment? Yeah.
So for the study in the prior slide, did that have to do anything with ghrelin, or just that was separate?
No, this has no ghrelin. This is a different study with no ghrelin.
Okay.
This is they just chemogenetically excited inhibited glutamatergic cells in ventral hippocampus, and exciting them causes less eating, which does kind of make sense, because if you broadly excite the whole ventral hippocampus, it causes more acute anxiety, and typically the mouse will eat less if it's very stressed out and scared and anxious. So this result does make sense with the anxiety results. And it was also kind of looked at which projections from the ventral hippocampus could be important for controlling feeding. So the ventral hippocampus projects very strongly to the lateral septum. Then they see that the optogenetic stimulation of this projection from ventral hippocampus to lateral septum decreases feeding. So that could be a projection through which this ventral hippocampus is acting to change feeding. And activation of these projections to the bed nucleus of the stria terminalis also seems to decrease eating. And the bed nucleus of the stria terminalis is a region that's very strongly interconnected with the amygdala, and has been mostly studied due to its role in defensive behaviors and anxiety and things like that. But there's also some emerging data showing the bed nucleus of the stria terminalis is important to control feeding. So they show that activation of this ventral hippocampus projection to bed nucleus of the stria terminalis decreases feeding. So both projection to bed nucleus and lateral septum decrease feeding. And activation of the cell bodies in general also seem to decrease feeding. Okay, so any questions about that one? Yep.
Is it decrease in feeding just because the diabetes is associated with feeding, or is it also because it induced anxiety?
It could be both. Yeah, because they didn't really control for anxiety here, which would be good because we know this region does cause more anxiety. And if you are more anxious, the mouse will totally eat less. That's very expected. Yep.
I know the general stimulation of the ventral hippocampus was only glutamatergic cells. Are these projections also exclusively glutamatergic, or do we have?
Yes, generally projection cells from hippocampus and cortex are glutamatergic. I mean, there's some small number of GABAergic cells that project from those areas to faraway regions, but it's overwhelmingly glutamatergic. But there are inhibitory projection cells in other regions. Like in the accumbens, all the projection cells are GABAergic, for example. And then speaking of accumbens, the next experiment is about that, which is they were seeing what happens to hippocampus projections to nucleus accumbens during these cocaine-related behaviors. So the nucleus accumbens, as you probably have heard, is a region that has been very extensively studied due to its role in motivated behaviors and drugs of addiction and depression and things of that sort that involve changes in reward circuitry. So the ventral hippocampus projects there. If you inject a virus in ventral hippocampus, you see a lot of axon terminals in accumbens. So they thought maybe this projection has a role in some drug-related behavior because the accumbens is so important for these drug-related behaviors. Okay, so what they did is they injected channelodrops in ventral hippocampus. Then there's some axon terminals that go to accumbens that have channelodrops in them. And then they patch clamped cells in the accumbens and activated optogenetically the axon terminals from ventral hippocampus containing channelrhodopsin. And the optogenetic activation leads to presumably release of glutamate. So you see this response appearing in black. And then that's the original response you see in black. So if you then put in AMPA receptor blockers, you see what's the NMDA response. And then if you put NMDA receptor blockers, you see what is the AMPA response component of that curve. And this is important because, as we talked about in the LTP class extensively, one major thing that happens after a projection gets potentiated or strengthened is that there is insertion of new AMPA receptors in those dendrites. So a common way to measure if some synapse has been strengthened is to measure if that synapse now has a bigger AMPA component than before potentiation, because you're inserting new AMPA receptors after synapse is strengthening. So now this synapse has more AMPA receptors. So the way that's measured is by measuring the size of the response you get for the AMPA component, which is this blue component, divided by the size of the response for the NMDA component. So this AMPA and NMDA ratio. So a synapse that has been strengthened compared pre-strengthening would typically show a higher AMPA and NMDA ratio because more AMPA receptors has been inserted into that dendrite and the NMDA receptor amount continues the same, so there should be a bigger AMPA and NMDA ratio, which would mean that in this case, if you do this experiment before and after, you see a bigger AMPA component, which is the dark green. So you see the dark green is bigger after the cocaine exposure, and the dark green is the AMPA component of response, which appears when you block the NMDA component. Because if you just excite the axon terminals, they release glutamate. The glutamate binds to both AMPA and NMDA receptors, so you see a mix of both responses. But then if you block one type of response but not the other, then you see emerging what the pure AMPA component, the pure NMDA component is like. So basically the AMPA component part of the response increases after the cocaine exposure. So it's like the take-home message is the projections from ventral hippocampus to accumbens gets potentiated after cocaine exposure, which means that synapse is now stronger because there is more AMPA receptors in the dendrites of those accumbens cells that receive that synapse. So you really see here how those studies that they did in brain slices in hippocampus with dorsal hippocampus doing LTP, and they found all those mechanisms. That's not just some strange thing that's happening in a petri dish, but those same effects seem to happen also in a real situation, which is a mouse before and after cocaine exposure. So this could have, if you think about it, lots of important clinical implications, because maybe the person that has exposure to cocaine also has a increase in synaptic strengthening in this pathway, and that could play a role in addictive behaviors, and that could feed into like gang activity and crimes and sociological effects. So the potential implications of these findings are kind of very broad and important. Okay, so they also tried to see if this projection had something to do with behaviors induced by cocaine. So if you just have a rat that's not doing cocaine, which is the normal rat, he is walking around, he's looking at stuff, he talks to the other rat, he rears up against the wall a little. Okay, but then if you put a bunch of cocaine in the rat, then the rat gets very hyper-locomotive. Then the rat is like, I can see every whisker of that other rat, and he's very hyper, and he's running around and does all sorts of things, very agitated. So it's kind of like if you look at the most famous cocaine movie of all time, Scarface, you'll see this, that in the beginning he's like a very reliable kind of businessman. He's like, no, I won't do my own cocaine. I have to separate doing cocaine business with consuming cocaine. But by the end of the movie, he's just like swimming in like hills of cocaine. And then when they invade his house, there's like this much cocaine on his desk. He's super coked up. And then he does all these things, like he shoots at like 500 people. He gets shot multiple times, and he still goes around, and then he falls in that pool, and then the cocaine rampage ended. But then what we see is that both in rats and Scarface, being all coked up causes hyperlocomotion. And then that's the thing that they're measuring here, but sadly they couldn't do a interesting home invasion cocaine swimming scene. So they did this, which is the next best thing, which is just inject cocaine in the rat, then they just run around a little more. Okay, so then they see if you inhibit this projection from ventral hippocampus to accumbens, they run around less. And then if you activate that projection, they run around more in the cocaine-treated mice. But here is the really cool result, which is if you activate or inhibit that projection in mice that did not do cocaine, then it doesn't cause a change in locomotion. So it's not that this projection just causes the mouse to run around more or run around less. It causes it to run around more or less only if the mouse had this cocaine exposure experience before. So this is a pretty cool result. Yep.
So they administer cocaine to the mouse and then optogenetically stimulate the pathway?
Yes.
Okay.
Yeah, and then we know so far the two things happen, which is that the synapse gets strengthened, and presumably this strengthened synapse is what now can control this behavior. Because the pre-strengthened synapse, which is here in the not cocaine control mouse, this synapse that has low AMPA and MDA ratio, if you activate and inhibit that, it doesn't cause more or less of the behavior. So it shows that there's some functional behavioral consequence of that synaptic strengthening that's happening here, which ties into the LTP studies that were done in slice many decades ago, where they found that potentiating the synapse in LTP causes more AMPA insertion. Yeah.
And then, like, obviously this has to connect with addiction.
Hmm?
This obviously connects with addiction.
Mm-hmm.
Because you strengthen it, it won't.
Yes.
But how does it necessarily go back to that over time and kind of create it more incentive?
Oh, this doesn't explain, like they didn't look into that, because it's a very complex phenomena, because there is the withdrawal, then there is the kind of relapsing again. There's a lot of parts in this addiction behavior. So here they didn't study that because this is not this hippocampal involvement in addiction is not some well-studied field. Because that's not what it's typically known for, So this is kind of a newer discovery. But those are probably things that they're studying at this moment. Like, is it, because here they showed it's important for the behaviors that happen right after some amount of exposure, and then there's the natural follow-up questions, which is probably what you were getting at. So what happens if you, in relapse of addiction, or what happens in the extinction of addiction? So there's a lot of things that probably the circuit is doing that they didn't look at. Yeah.
Professor, going back to the last slide, so you mentioned that after the cocaine exposure with the AMPA blocker, we get a strengthened synapse signal.
We get to what?
We get this strengthened synapse
Yes.
signal.
Mm-hmm.
But how does that make sense? Doesn't the LCB depend on AMPA receptors? I mean, both NMDA and AMPA receptors, but
Mm-hmm.
it leads to. Did the cocaine lead to more insertion of AMPA receptors?
Yes.
Oh, okay.
Yes. This cocaine caused a potentiation of this synapse from hippocampus to accumbens, which means the synapse is stronger by having more AMPA receptors in the dendrites.
And so the block didn't really do much because there was already so many AMPA receptors present.
No, what didn't do much?
The block, the AMPA blocker,
Oh, no. Oh, sorry. This is different experiment. So first the rat is there. Then you made it become exposed to cocaine. Then you made a slice of the brain which contains the accumbens. And then in that slice of the brain, you have some mice that were exposed to cocaine, some that were not. Of the mice that were exposed to cocaine, you see in those mice the synapse has more AMPA receptors. And the AMPA receptor component was found out in the patch clamping by adding either the NMDA blocker to see the AMPA response or by adding the AMPA blocker to see the NMDA response.
I see. Thank you very much.
Yeah, so this is an important concept, which is basically if you excite those terminals, you see this black response, which is the total excitation the ventral hippocampal cells produce when they release glutamate on the accumbens. Then you're like, okay, this glutamate response is caused by AMPA receptors and NMDA receptors, so which one is causing what amount of the response? So if you repeat the experiment in the presence of the AMPA blocker, which is in red, then the AMPA receptors are blocked, so the remaining response is the NMDA response, which is the red. And then conversely, if you put the NMDA blocker, you see the AMPA response, which is the blue.
Thank you.
Yeah, and then you measure the ratio of the two responses, which is this AMPA and NMDA ratio. So typically, after strengthening potentiation, there is more insertion of AMPA receptors, there is bigger AMPA and NMDA ratio.
Thank you.
Yeah, welcome. Yeah.
I think a quick clarification question on the next slide. So the orange, would that be chlorodiopsin for inhibition?
Yes.
And blue is excitation.
Yeah, inhibition in orange, excitation in blue. Yeah, so here you see a bidirectional control. So it's as if this projection before strengthening, when it has fewer AMPA receptors, this projection didn't seem to have anything to do with locomotion. But after synapse gets strengthened, it becomes strong enough that now it can drive this locomotion, which is why exciting or inhibiting it changes the locomotion. Yep.
So this is why some sort of silent synapses in the pathway?
It could be that that's there, but the thing that they measured was this AMPA receptor insertion, which they didn't measure it in silent synapses because that's a very specific protocol where, that could be happening, so it's totally possible that happened. And unsilencing the synapse will also lead to an increase in AMPA receptor component. So that might be happening, but what they measured were just pre-existing, not silent synapses that now they have more AMPA receptors. Because to measure the silent synapse, you have to do that thing of decreasing the stimulation amount a lot until you see no response, and then you have to depolarize the cell, then find a response. And so they didn't do that kind of patch clamping here. But that could happen. Like tau synapses are being unsilenced in brain circuits when things are being learned. Okay, so they also saw that stimulation of the projection from ventral hippocampus to accumbens increased instrumental behavior. So here the experiment is there's a lever, and then when the mouse presses the lever initiates activation of the projection from ventral hippocampus to accumbens. So the idea here being if they like or are motivated or is rewarding, nice, interesting, etc., to receive the stimulation, then they will do more lever pressing to receive the stimulation. And indeed, you see they do more lever pressing to get this optogenetic stimulation, suggesting that the stimulation is rewarding, good, positive, something of that sort. Which makes sense because these drug pathways are usually positive, nice, rewarding, etc. Okay, so yeah, that was the end of our slideshow. And then what we showed in conclusion is that hopefully you can appreciate how the kind of knowledge has really progressed, because we started out with how is memory encoded in the brain. Maybe it's like animal spirits make little holes in the brain tissue, and then the tissue has some holes in it, and that holds the memory. And then we had some people that didn't do experiments but just came up with some conceptual ideas that were many decades later found to be true experimentally. And then there were some people that studied kind of brain slices in vitro in a petri dish and found some mechanisms that cause synapse strengthening and weakening and etc. And later other people found out that those same mechanisms were happening in real animals learning real things, and the effects were kind of very large, right? So in the Morris-Water maze experiment, we saw that just knocking out one receptor, which is the NMDA receptor in one region, which is CA1 dorsal hippocampal region, just doing that one manipulation made the mouse completely incapable of remembering where the hidden platform was in the bucket of water, Morris-Water maze experiment. So a very minor change can completely abolish all of this spatial learning. And then now we're seeing a hippocampus has also this ventral part, which is not involved in spatial learning and memory. It's involved with these more emotional behaviors, like being more or less anxious, or eating more or less, or doing cocaine and so forth. And more interestingly, we see that the same mechanisms that were found in LTP in the slice, for example, are now found in this very different pathway going to the accumbens and in some real experience, which is not LTP, but it's a real mouse that did real cocaine. And the same mechanism that was seen in the Petri dish in LTP seem to be happening here. So that's also the last thing I want to just make clear, which is that LTP and LTD are not things that really happen in animals. Those are weird lab protocols that happen in brain slices with a human putting an electrode in the brain slice. So you don't have an LTP. But the reason why the LTP is important is that those weird experiments strengthen and depress synaptic pathways using kind of biophysical, biochemical mechanisms that are clearly being used in real situations on the animal, is actually learning something, forgetting, changing its behavior, etc. Yeah.
So if I'm understanding, we, like in living animals, they have mechanisms that are analogous to LTP that we do in a lab.
Yeah, not just analogous, but often the same. Like it's the same AMPA receptors being inserted. It's not like, oh, one other excitatory receptor is inserted. It's like, so it's like the same things happening. But it's just it's more that the induction of that is happening through a real experience rather than a electrode doing a strange electrical thing in the brain slice. Okay, you got it? Okay, well, so yeah, thank you everyone for coming, and then the final exam will be on Tuesday, and there was also some controversy on what will be on it. So because the syllabus says that only material covered in the lecture will be on the exam, then only material covered in the lecture will be in the exam. So this paper is in the discussion. Also, the first paper we talked about, this paper is also in the discussion, but it was covered in lecture, so this would also be in the exam. Okay, so thank you everyone for coming, and I hope you enjoyed learning. Thank you.